In the next lecture, we will see the basics of the OECD and the Good Laboratory Practice, as well as the mutual acceptance system. What is the OECD? The OECD is an organization for economic cooperation and development which is an intergovernmental organization of approximately 40 member countries, founded in 1961 to stimulate economic progress and world trade. This is a residue of the Marshall Plan. Cited, it is a forum and its members are countries which describe themselves as committed to democracy and the market economy, providing a platform to compare policy experiences, seek answers to common problems, identify good practices, and coordinate domestic and international policies of its members. The headquarters is in Paris. The OECD is recognized as a highly influential publisher of mostly economic data through publications as well as annual evaluations and rankings of member countries. Here on the map you can see the member countries, especially in the enlargement in Europe. The OECD publishes books, reports, statistics, working papers, and reference materials. All titles and databases publishes since 1998 can be accessed via OECD iLibrary. All the other libraries and archives dated from 1947. External researcher can consult OECD publications and archival materials on OECD premises by appointment. The reference work. The OECD is responsible for the OECD guidelines for the testing of chemicals, which is a continuously updated document that is a de facto standard in most countries, a so-called soft law. It has published the OECD Environmental Outlook to 2030, which shows that tackling the key environmental problems we face today, including climate change, biodiversity loss, water scarcity, and the health impacts of pollution, is both achievable and affordable. The neutrality, this is my own opinion, is disputed or may be disputed. Testing chemicals in different languages can be read as you see, and here is the basic work of the good laboratory practice and compliance monitoring. And here you see the translation from basic science to human studies, the process, the workflow from basic science research, translation to human, translation to patients, translation to practice, and translation to community. So this is the workflow, and you see that there is a lack of data and technology for the translation from basic science to human studies. In the slogan, it is called that this is the valley of death. The classical drug development process is composed of 4 well-defined stages. Stage 1 is the discovery of potential new drug products, chemical compound, for example, not covered by a regulatory standard. This is stage 1. So you see that this is highly important, that from the basic science should be a translation to the preclinical phase. It means that from in vitro and ex vivo stage to animal models. From animal models, the next stage is the clinical trials, phase 0, 1, 2, and 3. And after we arrive to the approved therapy phase. So, to see an example that we have in the cell type, in the basic science, we have in the cell types different cells which can be used in vitro science. In vitro science, we use them in vitro and ex vivo studies. Later on, in the preclinical phase, in vivo studies using small and large animal models. This is an iterative process, the basic science and the preclinical science. After comes the translation. This is the clinical trials, injectable human therapy, for example. Here the example is something to improve the bone development. The next figure shows us that the resources are for the development are so-called sufficient in the academic environment and industrial environment. In other words, in the field of the basic science and clinical science. But there is a valley of death concerning the resources, concerning the translational science, which includes reproducibility, clinical relevance, structural, investigator base, technical expertise, support, intellectual support, regulatory, privacy issues, funding, incentive risks, collaboration, follow through time. So this is a problem in the development pipeline. You see that for this valley of death we have various solutions. For example, by funding, in case of, for example, in the United States, the National Institute of Health can give funding. And in the clinical development, mostly large pharmacological companies can provide funding. Okay, we arrive to the stage 2 of the medicine development. This is non-clinical, not in human. It includes safety testing, and the GLP principles are specific for this stage. It means that the toxicology, the safety pharmacology, the pharmacokinetics, and the bioavailability. And the next stage is the stage 3, the clinical studies in human. This is the good clinical practice is the basis for quality standards, ethical conduct, and regulatory compliance. And the standards are described in the ISO system. The so-called phase 1 study the tolerance of test drugs, define human pharmacokinetics, to phase 2, which studies the dose-effect relationship, and the phase 3, it is a full-scale, often multi-center clinical efficacy trials in patients. And the stage 4 is the post-approval stage. The drug is registered, available on the market, and any subsequent clinical trials, the so-called phase 4, must comply with the good clinical practice and the standards written in the ISO, the national standard. From stage 4 of development and continuing throughout the rest of the drug's lifetimes, good manufacturing practice applies to all manufacturing of bulk and formulated product. So the good manufacturing practice is described in the WHO standards, the so-called Geneva document 1999. Why GLP? GLP, good laboratory practice, is to decrease the influence of other than test material influence on the results. Why are the OECD principles of good laboratory practice needed? In the history in 1970s, prior to the introduction of the GLP, some governments discovered fraudulent studies that had been submitted by testing laboratories to regulatory authorities. As a result, OECD governments decided that there would be value in developing a set of principles applied across all OECD countries concerning the generation of quality test data. This would ensure that before making regulatory decisions concerning the safety of chemicals that will enter the market, governments would have confidence that the data upon which they make their decision are valid and of high quality. Through standards built into the OECD principles of good laboratory practice, which allow the traceability of studies, the good manufacturing practice inspectors who visit test facilities can audit the results of a study long after it has been completed. As the application of good laboratory practice is harmonized across the OECD countries, governments can accept data from other countries with the assurance that it will be valid and of high quality. See later mutual acceptance of data system below. Good laboratory practice is a quality system concerned with the organizational process and conditions under which non-clinical health and environmental safety studies are planned, performed, monitored, recorded, archived, and reported. And here you see a copy of the OECD papers. figure shows us a timeline of good laboratory practice guidance from FDA, Organization for the Economic Cooperation and Development, the International Society for Pharmacological Engineering, ESPE, Good Automation Manufacturing Practice, GAMP, and the United Kingdom's Medicine and Healthcare Products Regulatory Agency, MHRA. GXP is an abbreviation for good practices. So you see that a good laboratory practice was introduced 1979, and after the OECD series on principle of good laboratory practice and compliance monitoring, number 15, 2007, and all the mentioned authorities, and today we are OECD draft advisory document of the working group on good laboratory practice on GLP data integrity 2020. So this is a never-ending story. So the good laboratory practice GLP has in the center the element of good laboratory practice. You see that this is a quality system of management controls for research labs and organizations. The elements are the reagent, the materials, and the certifications, the accountability, the statistical procedures, the instrumentation validation, documentation and its record, quality assurance, specimen and sample tracking, certification of analysis, certification of lab facilities. And here you see a copy of the part 1 of the OECD principles of the GLP with the introduction, preface, and section 2, test facility organization and personnel, quality assurance program, facilities, and the continuation apparatus, material, and reagent. The number 5 test systems included physical, chemicals, and biological. 1. 6 test and reference items, receipt, handling, sampling, and storage, and characterization. Number 7 is highly important, the SOPs, the standard operating procedures. Number 8, performance of the study with study plan characteristics. Number 9, about the reporting of study result. And number 10, storage and retention of records and materials. There is a list of definitions in this OECD documents giving explanation of the test site management, sponsor, study director, which is a central figure, central person in the GLP, principal investigator, quality assurance program, and the standard operating procedures, which mean documented procedures which describe how to perform tests or activities normally, not specified in detail in study plans or test guidelines. A good laboratory practice in laboratories for non-clinical safety testing. A set of standard applied to non-clinical environmental health and safety studies is the principle of GLP. This is a set of standard. The principle of GLP is a formal term. This is a regulatory document. There is an international harmonization in the field of OECD principle of good laboratory practice. The OECD formulated the first worldwide OECD principles of GLP 1981, revised in 1998, to avoid non-tariff barriers to trade, to promote mutual acceptance to non-clinical safety tests, and to eliminate unnecessary duplication of experiments. So in this way, this is part of the 3 R's of the animal protection, the reduction of the number of animals involved in the study. What are the OECD principles of good laboratory practice? First of all, the principles of good laboratory practice are a managerial quality control system covering the organizational process and the conditions under which non-clinical health and environmental studies are planned, performed, monitored, recorded, and reported. The OECD principles of GLP are followed by test facilities carrying out studies to submitted to national authorities for the purposes of assessing the health and environmental safety of chemicals and chemical products, which may also be of natural or biological origin, and in some circumstances may be living organism. Depending on the jurisdiction, the principles of GLP can also be applied to non-clinical safety testing of other regulated products, such as medical devices. And the second slide about the principles of the good laboratory practice, it defines, the principles defined the responsibilities of test facility management, study personnel, and quality assurance personnel that are operating within a GLP system, and define minimum standards concerning the suitability of facilities and equipment to perform studies, the need for standard operating procedures, SOPs, documentation of raw data, study reports, the archiving of records, etc. So you see that this official citate can be given in a little bit easier way. OECD principles of GLP define and describe a quality system concerned with the organizational processes and conditions under which a non-clinical health and environmental safety study is conducted. It is easier, but not a lot. Non-clinical laboratory study means in vivo or in vitro experiments in which test articles are studied prospectively in test systems under laboratory conditions to determine their safety. So you see that no clinical environment and conditions. So continuing the principles, these are this is a quality system concerned with the organizational processes and the conditions under which non-clinical health and environmental studies are planned, performed, monitored, recorded, archived and reported. But it does not concern with the technical validity of the study. It's highly important. It does not concern with the technical validity of the concern. For example, failure during the measurement. The first question is, which arrives, that what is a test facility? A test facility includes the persons, premises, for example, a testing laboratory, and operational units that are necessary for conducting a non-clinical health and environmental safety study. See below. For multi-site studies, those which are conducted at more than one site, the test facility comprises the site at which the study director is located and all individual test sites, which individually or collectively can be considered to be test facilities. So everything practically. Okay, fundamental points of the GLP are the GLP applied in whatever industry targeted stresses the importance of the following main points: resources, rules, characterization, documentation, and quality assurance. In details, resources, organization, personnel, facilities, and equipment. Rules, protocols, SOPs, concept of study director. Characterization, test items, test systems, animal models. Documentation, raw data, final report, archives. And quality assurance, independence from study conduct, and separation physically and by organization ensures that different functions or activities do not interfere with one another. Important in pharmacy and dose mixing areas to separate histopathology or analytical laboratories and the animal facility. You have to minimize the effects of environmental variables on the animals facility designed and operating to prevent animals coming in contact with the disease and with the test item other than the one under investigation. Okay, so this is how the GLP of the animal facility, this separation. The rules are written in the protocols. The principal step of studies have to be described in a study protocol or study plan. The protocol or study plan has to be adapted by the study director through dated signature before study starts, and alterations to the study design cannot be made unless by formal amendment procedure. The written procedures are routine procedures described in standard operating procedures, SOPs. Standardization of certain techniques to facilitate comparison of results. Procedures must be reviewed regularly and modified if necessary to reflect the actual state of the art. Standard operating procedures must be available at workplace and in current version. So For example, SOP for mice and rats should include origin and genotype of the animals, keeping conditions like temperature, humidity, bedding, and environmental enrichment, background noise, feeding, drinking, the caretaking, the health control, prevention and alleviation of pain and suffering, and, last but not least, the record keeping. The characterization include preclinical safety testing of pharmaceutical compounds, requires detailed knowledge about the properties of the test items and of the test system, which is often animal, to which it is administered. Identity, purity, composition, stability, impurity profile should be known for the test item, for the vehicle, and for the reference material. If the test system is an animal, it is essential to know such details as its strain, health status, and normal biological value. What types of tests are carried out at such facilities under good laboratory practice? The OECD principles of good GLP concern non-clinical testing of chemicals or chemical product examined under laboratory conditions or in the environment, including work conducted in greenhouses and in the field. They do not include studies which use human subjects. Examples of studies carried out under GLP include inter alia, from 1 up to 8, physical chemical testing, toxicity studies, and lot, a series, mutagenicity studies, environmental toxicity studies on aquatic and terrestrial organisms, studies on behavior in water, soil, and air, bioaccumulation, studies to determine pesticide residues in food and animal feedstuffs, studies on effects on mesocosm and natural ecosystems, and last but not least, analytical and clinical chemistry testing. And what type of chemicals and chemical products are covered under the OECD principles of GLP? It is a list of 7 items. First, pharmaceutical products. Second, pesticides including biocides, cosmetic products, veterinary drugs, food additives, feed additives, and industrial chemicals. Depending on the jurisdiction, the principle of GLP may also be applied to non-clinical safety testing of other regulated products such as medical devices, as mentioned before. What about the documentation, the archives? For reasons of reconstruction many years later, safekeeping of all records kept in integer state and can neither be lost nor altered. Restrict access to archives to a limited number of people and maintain records of login and logout for both documents and people. Quality assurance. Quality assurance defined by GLP, a team of persons charged with assuring the management that GLP compliance has been attained in the test facility as a whole as well as within each study. Quality assurance has to be independent of the operational conduct of the studies, and it functions as witness to the whole preclinical research process. What is the ISO accreditation versus GLP? A fundamental requirement of the GLP principles not covered in ISO or EEC, the use of study plans and study director as a concept. This is a special feature of the GLP. More stringent under the GLP, the recording and reporting of data, management of data retained in archive to allow complete reconstruction of study, and the program independent quality assurance, including internal audits of every study. And now comes a question and answers concerning the GLP. How do governments know that a study conducted at a test facility was carried out according to the OECD principles of good laboratory practice? We have two answers. The OECD countries in which non-clinical health and environmental safety testing is carried out according to the OECD principles of good laboratory practice have established national GLP compliance monitoring program, CMP, with responsibilities for monitoring GLP compliance of test facilities within their territories. GLP compliance is verified by compliance monitoring programs through inspections of GLP test facilities and audits of GLP studies. A test facility which has been subject to periodic inspections by the compliance monitoring program and found to be operating in compliance with the principle of GLP is recognized as a GLP compliant facility. And how can test facilities become recognized as GLP compliant? Any test facilities that conduct non-clinical health and safety studies, for example, a university, research institute, private enterprise, governments, etc., can become OECD GLP compliant or recognized. This includes facilities in OECD member countries as well as non-OECD economies who become full adherent to the Mutual Acceptance of Data (MAD) system, see below. In most countries, facilities that wish to become recognized as GLP compliant can apply to the government CMP. The CMP then conducts an inspection to determine if the facility complies with the OECD principles of GLP. In other countries, CMPs can inspect any test facility claiming to conduct studies according to GLP. Are the OECD principles of GLP established in and revised in 1997 still relevant today given advances in testing methods? Yes, the OECD principles of GLP make up a general framework concerned with the process a test facility uses to conduct a test, not a specific substance being tested, nor the scientific approach followed when carried out a test. The OECD principles of GLP do cover all types of tests, including those not envisioned when the principles were first established. OECD does periodically publish advisory documents which provide additional guidance or clarity concerning the application of GLP to new testing approaches. For example, guidance for in vitro test methods and deleting some animal tests. And finally, what is the OECD's Mutual Acceptance of Data (MAD) system? Why was the MAD system established? As many of the same chemicals are produced in more than one country or are traded across countries, different national chemical control policies can lead to duplication in testing and government assessment, thereby wasting resources of industry and government alike. Differences in regulations and test standards discourage research, innovation and growth, and they increase the time it takes to introduce a new product onto the market. They can also lead to inefficiencies for governments because authorities cannot take full advantage of the work of others, which would have reduced the resources needed for chemical control. By reducing the application and creating a framework for the sharing of work, the mutual acceptance of data system saves governments and industry a lot of money and save a lot of animal life. This is part of the 3 R's, reduction of the animal involved in animal experimentation. The primary aim of the MAD system is to help governments protect human health and the environment. As other benefits occur, fewer duplicative tests means there is a reduction in both costs and the number of laboratory animals needed. As a result of the harmonization of test methods and GLP, governments are able to collaborate on assessment of the same chemicals and share the burden of assessing the thousands of chemicals which are on the market. As a result, governments can assess chemicals faster and more cost-effectively. For example, the OECD's Hazard Assessment Program, which uses data generated according to OECD test guidelines and GLP. Countries have used these assessments for their national chemical management activity, for example, for classification and labeling. The OECD Mutual Acceptance of Data (MAD) system is a multilateral agreement which allows participating countries including non-member economies, to share the results of various non-clinical tests done on chemicals. Under MAD, a non-clinical chemical safety study developed using OECD test guidelines and OECD principles of good laboratory practice in one member country or non-member full adherent must be accepted for assessment purposes in all member and adhering countries. This is the concept of testing once, accepted for assessment everywhere. And here an illustration of the countries of the OECD Mutual Acceptance of Data System. The OECD is red and the adherent countries are pink. The legal framework for the MED system. The multilateral agreement is composed of 3 OECD Council acts adopted by OECD ambassadors. Number 1. The 1981 Council decision on the mutual acceptance of data in the assessment of chemicals, revised in 1997, that states that test data generated in any member country in accordance with OECD test guidelines and OECD principles of good laboratory practice shall be accepted in other member countries for assessment purposes and other uses relating to the protection of human health and environment. So this is the legal formulation what I explained before. Number 2, the 1989 Council decision recommendation on compliance with principles of good laboratory practices, which establishes procedures for monitoring GLP compliance through government inspections and study audits, as well as framework for international liaison among monitoring and data receiving authorities. And number 3, the 1997 Council decision on adherence of non-member countries to the Council acts related to the mutual acceptance of data in the assessment of chemicals, that sets out a stepwise procedure for non-OECD economies to take part as full members in this system. How does one government participating in the MED system have confidence in the quality and integrity of data generated in another country in the system? The OECD Working Group on GLP has established a program of on-site evaluations of each country in the MED system. Every national GLP compliance monitoring program is subject to a regular on-site evaluation visit, normally every 10 years. A team of 2 or 3 representatives from other GLP compliance monitoring program, CMP, participate in the site visit. How does non-member economies join the MED system? The non-member economies government confirms that it will provisionally adhere to MED. It agrees to accept, for assessment purposes, non-clinical data generated in the testing of chemicals with OECD test guidelines and OECD principles of good laboratory practice from countries adhering to the OECD Council Acts and MAD. OECD countries and full adherents are not obliged to accept data for assessment purposes from the provisional adherent. As the provisional adherent is setting up a GLP CMP, it is invited to attend OECD working group on GLP meetings and training courses, and to serve as an observer in on-site evaluations of other countries. Once the provisional adherent has fully developed its GLP CMP according to OECD guidance, an on-site evaluation visit is carried out. The country becomes a full adherent to MET system. And finally, the last slide, what is a full adherent non-member economy? A full adherent is a non-member economy whose GLP compliance monitoring program, CMP, has been evaluated by OECD and complies with the OECD Council Acts and Mutual Acceptance Data System. As a result, non-clinical health and environmental safety testing of chemicals and chemical compounds conducted in a full adherent in accordance with OECD test guidelines and OECD principles of good laboratory practice shall be accepted for assessment purposes in OECD member countries, provisional adherents and other full adherents. Argentina, Brazil, India, Malaysia, Singapore and South Africa are full adherents. And to finish with, thanks for your attention, and you see this pile of books. These are similar to the OECD guideline. There are more and more, and one is deleted, one is replaced. So please, your eyes keep on the refreshment continuously. Once more, thanks for your attention.